Hello everyone, welcome to the channel Microbial World. In this video, we will be learning about Escherichia coli. E. coli was discovered by Theodore Escherichia in 1885 after isolating it from the feces of newborn. E. coli is the normal flora of the human body, which means this bacterium is commonly found living in a specific part of the human body without causing any harm. The primary habitat of E. coli is the gastrointestinal tract of humans and many other warm-blooded animals. They belong to the family Enterobacteriaceae. Most of them are harmless and opportunistic. When we say E. coli are opportunistic, it means that they normally do not cause any disease, but it can lead to infections when it takes advantage of certain conditions, such as when the immune system is weak, or if the microorganism is introduced into a part of the body where it is not normally found. For example, E. coli moving from the intestine to the urinary tract, then this can lead to infections. And also if there is changes in the normal microbial community, like due to antibiotic use, then this can also allow the opportunistic pathogens to proliferate and cause disease. Morphology. E. coli is a gram-negative rod-shaped bacteria. It is 1 to 3 into 0.4 to 0.7 micromuem in size and 0.6 to 0.7 micromuem in volume. It is arranged singly or in pairs. It is motile due to peritrichous flagella. Peritrichous flagella is a flagellar arrangement where multiple flagella are distributed all around the cell surface. So this numerous flagella will allow E. coli to move in a coordinated manner. And there are also some strains that are non-motile. Some strains may be fimbriated. Fimbriae are hair-like appendages that is found on the surface of the bacteria. It allows bacteria to adhere to surfaces such as host tissues. Some strains have a polysaccharide capsule. Capsule is a thick gelatinous layer that surrounds the cell wall. Now mainly E. coli strains that is isolated from extraintestinal infections such as the bloodstream, urinary tract, these strains will have polysaccharide capsule. They are non-sporing, so E. coli do not form bacterial endospores. Antigenic structure. The key components of antigenic structure of E. coli are lipopolysaccharide and O antigen, capsular antigen, flagellar antigen, and fimbrial antigen. Lipopolysaccharide is a major component of the outer membrane of gram-negative bacteria like E. coli. It consists of three parts: lipid A or endotoxin, core oligosaccharide, and O-specific polysaccharide or O antigen. Lipopolysaccharide, it contributes to the structural integrity of outer membrane and thereby it serves as a barrier against host defenses. O antigen or somatic antigen. It is the outermost part of the lipopolysaccharide. And this O antigen, it is highly variable among different strains of E. coli. It is used in serotyping to classify E. coli into distinct serogroups. For example, O157, O111, and this classification is based on the sugar composition and glycosidic linkages. Flagellar antigen or H antigen. Flagellar antigen contributes to motility and serves as antigen. H antigen, it is located on the bacterial flagella and it is also used in serotyping to further differentiate strains within O serogroup. Similar to O antigen, H antigen also exhibits variations and this allows to classify E. coli strains to different H types. Capsular antigen or K antigen. As we have already said, some strains of E. coli produce a polysaccharide capsule surrounding the cell wall. So this K antigen, it contributes to virulence. Virulence means it is the ability of a pathogen to cause disease and the resulting severity of the illness. And they also resist phagocytosis by host immune cells. So what is phagocytosis? Phagocytosis means certain cells of the immune system has the ability to engulf and digest foreign particles such as bacteria, debris or dead cells in order to eliminate them from the body. It is called phagocytosis. So this K antigen will contribute to virulence as well as they will resist phagocytosis by host immune cells. And K antigen, it is less commonly used in routine serotyping when compared to that of O antigen and H antigen. F4 fimbrial antigen are heat labile proteins, which means they are heat sensitive, and it is present in the fimbriae. Cultural characteristics. E. coli is a facultative anaerobe, which means it can grow in the presence and absence of oxygen. Its optimum growth temperature is 37 degrees Celsius and it ranges from 10 degrees Celsius to 40 degrees Celsius. On nutrient agar, they appear as large, circular, low convex, greyish white, smooth and opaque colonies. On blood agar, the colonies are big, circular, greyish and moist. Non-hemolytic colonies, that is gamma hemolysis or complete lysis of red blood cells, that is beta hemolytic colonies, are formed on the blood agar. On McConkey agar, the colonies are circular, moist, smooth and of entire margin. They appear as flattened pink and they are lactose fermenting colonies. On eosin methylene blue agar, green metallic sheen colonies are formed. On M. endo agar, colonies are green metallic sheen. They metabolize lactose with the production of acid and aldehyde. And on liquid media, E. coli shows turbid growth within 12 to 18 hours. Pathogenesis. The 5 E. coli pathogenic strains that cause intestinal infections or gastroenteritis that leads to diarrhea are enterotoxigenic E. coli, enteroaggregative E. coli, enteroinvasive E. coli, enteropathogenic E. coli, and enterohemorrhagic E. coli. Enterotoxigenic E. coli. It causes travelers' diarrhea. It is a gastrointestinal illness that affects travelers, mainly when they are visiting countries or regions that have poor sanitation. It is caused due to the ingestion of water or food that is contaminated with bacteria, viruses, or parasites. It is mainly observed in children of younger ages. The site of infection is small intestine. The source of the infection is fecal contamination of food or water. Symptoms include watery diarrhea, abdominal cramp, and sometimes nausea and vomiting. 2 types of enterotoxin is produced by enterotoxigenic E. coli. Heat labile toxins, labile toxin 1 and labile toxin 2. Heat stable toxins, stable toxin A and stable toxin B. So labile toxin 2, it is not related to human diseases. Labile toxin 1 is an AB toxin. It consists of 1 subunit of A and 5 subunit of B. So the B subunit will bind to receptors such as GM1 ganglioside. GM1 ganglioside is a glycosphingolipid that is found in the plasma membrane of various cell types including epithelial cells. So B subunit will bind to GM1 ganglioside and other glycoproteins that is present on the epithelial cell. Whereas A subunit will bind to GS protein. GS protein, it is also known as stimulatory G protein. It is an important component of the intracellular signaling pathway mediated by G protein coupled receptors. And GS protein will control adenylate cyclase. So this interaction will result in an increase in cyclic AMP levels, that is cyclic adenosine monophosphate levels. And it will result in more secretion of chloride and the absorption of sodium and chloride is decreased. This will lead to watery diarrhea and it will also trigger the secretion of prostaglandin and produce inflammatory cytokines that will result in further water loss. Now in case of stable toxin, heat stable toxin A is a small peptide. It interacts with transmembrane guanylate cyclase receptors and that will lead to the secretion of CGMP, cyclic guanosine monophosphate, and this will cause fluid hypersecretion. Fluid hypersecretion means it is the excessive secretion of fluids by cells or tissues in the mucosal surfaces of organs such as gastrointestinal tract or respiratory tract. This condition can lead to increased fluid accumulation in these organs and disrupt the normal physiological functions. Enteropathogenic E. coli. It causes infant diarrhea. The site of infection is small intestine. Symptoms include watery diarrhea, fever, and vomiting. So, how do they cause infection? Initially, the bacteria attaches to the small intestine epithelial surface cells and they destroy the microvilli. Microvilli are small finger-like projections that extend from the surface of the epithelial cells, particularly the cells lining the intestine and kidney tubule. So the bacteria first attaches to the epithelial cells in the small intestine and they destroy the microvilli and produce a lesion on the microvillus known as attachment or effacement lesion. Now this enteropathogenic E. coli has hair-like appendages on its surface known as bundle-forming pili. So this bundle-forming pili helps the bacteria to adhere to the epithelial cells lining the intestine, and then they form microcolonies on the epithelium surface cells. Attachment is carried out by genes present on the pathogenicity island known as locus of enterocyte effacement. So this pathogenicity island, it is a genetic region in the genome of pathogenic bacteria that contains a cluster of genes encoding virulence factors. So this locus of enterocyte effacement, it consists of more than 40 genes that can carry out attachment and destruction of host cells. Bacterial type 3 secretion system secretes active proteins in the host cells. One protein called TIR, translocated intimin receptor, is inserted on the epithelial cell. So once inside the host cell, this TIR will act as a receptor for another bacterial protein called intimin. Now this intimin, it is encoded by a gene that is located on the locus of enterocyte effacement. So this intimin, it will bind specifically to the TIR receptor. The binding of TIR receptor and intimin will result in actin polymerization and cytoskeletal elements accumulation and ultimately cause cell death. Enteroaggregative E. coli. It causes persistent watery diarrhea along with dehydration in infants. The site of infection is small intestine. The main characteristic of the bacteria is autoagglutination. It has autoagglutination adherens fimbriae 1, and this fimbriae will help the bacteria to adhere to the surfaces. So they will attach to the intestine surface. Now when the enteroaggregative E. coli, they attach to the intestinal epithelial cells, they show a stacked brick appearance. Okay. So they will attach to the intestine surface and stimulate mucus secretion, and that will lead to the formation of biofilm. And this biofilm will protect the aggregated bacteria from antibiotics and phagocytic cells. Now the toxin produced by enteroaggregative E. coli is called enteroaggregative heat stable toxin and plasmid encoded toxin that induces secretion of fluid. Enterohemorrhagic E. coli. The site of infection is large intestine. The cause of infection is consumption of undercooked beef or other meat products, unpasteurized milk, or uncooked vegetables. This can lead to infections. Symptoms include mild diarrhea to hemorrhagic colitis. Hemorrhagic colitis can result in bloody diarrhea known as dysentery, sharp abdominal pain, and vomiting. Enterohemorrhagic E. coli is related to hemolytic uremic syndrome. Now, hemolytic uremic syndrome is a very serious condition that is characterized by a combination of acute renal failure, low platelet count, that is thrombocytopenia, and hemolytic anemia, that is the destruction of red blood cells. Enterohemorrhagic E. coli serotype O157H7 is a common strain that causes disease. Enterohemorrhagic E. coli produces Shiga toxins, Shiga toxin 1 and Shiga toxin 2. Both are AB type toxins. Now let's see how it causes infection. The B subunit will bind to a specific glycolipid on the host renal endothelial cell and intestinal villi. This glycolipid is called globotriacylceramide, GB3. The A subunit, it is internalized and it is cleaved into two molecules. One part will bind to 28S rRNA and it will stop protein synthesis. Now, enterohemorrhagic E. coli, it is more pathogenic if it has both Shiga toxin activity and attaching and effacing activity. Enteroinvasive E. coli. The site of infection is large intestine. Few serotypes of enteroinvasive E. coli that are pathogenic are O124, O143 and O164. Now, how does the bacteria act? The bacteria invade and cause destruction in the colonic epithelium. Bacterial invasion is mediated by PINV genes. Symptoms include watery diarrhea that can progress into dysentery, fever, abdominal cramp, and blood and leukocyte in the stool sample. The bacteria cause lysis of phagocytic vacuoles and multiply in the cytoplasm. And these bacteria also form actin tails. Actin tails are structures produced by actin-like filaments in certain pathogenic bacteria. These actin-like filaments are structural proteins. So the formation of actin tails, it will help the bacteria to move within the cytoplasm and across the neighboring epithelial cells. Colonic ulceration can occur. This occurs due to epithelial cell destruction with inflammatory cytokines stimulated by bacteria. Clinical features. Gastrointestinal infections. As we have already discussed, it can result in watery or bloody diarrhea, vomiting, nausea, low-grade fever, dehydration and abdominal cramps. Urinary tract infection. It can cause dysuria, that is painful urination, increased frequency and urgency to urinate, hematuria, blood in urine, and flank pain in case where kidney is involved. Neonatal meningitis. E. coli can cause meningitis in newborn. It is either acquired through birth or maternal transmission. Symptoms include fever, poor feeding, irritability, seizures, and altered mental status. Septicemia. E. coli bacteremia can lead to septicemia, that is bloodstream infection. The symptoms include fever, chills, hypotension, systemic inflammatory response syndrome. This is a generalized inflammatory state that can cause widespread tissue damage and organ dysfunction. Diagnosis. Immunoassay and nucleic acid detection methods are used to determine the toxins and their associated genes. This is a very expensive method. For gastrointestinal infections, stool culture or PCR is carried out. Fresh stool sample is collected and cultured on selective media that promote the growth of E. coli. PCR allows a rapid detection and identification of specific genes associated with pathogenic strains. For example, genes encoding virulent factors like Shiga toxin in enterohemorrhagic E. coli. In urinary tract infection, urine culture is done. Midstream or catheterized urine sample is collected and cultured on appropriate media. Blood culture is performed to identify E. coli septicemia. Blood sample is collected from peripheral vein or central lines and it is cultured on blood culture bottles. If there is growth of E. coli in the blood culture bottles, then it indicates a bloodstream infection. For suspected meningitis, cerebral spinal fluid analysis can be done. Lumbar puncture is performed to collect CSF. Serological tests are used to detect antibodies against specific E. coli antigens or toxins. Treatment. Rehydration and supportive measures are taken. Hemorrhagic colitis and hemolytic uremic syndrome are treated by using hemodialysis or hemapheresis. Hemodialysis is a medical procedure used to remove waste products and excess fluid from the blood when kidneys are unable to perform these functions. Hemapheresis is a blood purification therapy. It removes specific components from the blood while returning rest of the blood components to the patient. To treat diarrhea caused by enterotoxigenic E. coli, enteroinvasive E. coli, and enteropathogenic E. coli, Trimethoprim or sulfamethoxazole, doxycycline or quinolones are used. Thank you for watching.